So this is part 4 of the biochemistry of human milk, and we will start off by talking about epigenetics. Epigenetics is the study of changes in organisms caused by modification of gene expression rather than alteration of the genetic code itself. It looks at the cellular layer that rests on top of the genome and the mechanism behind how genes are switched on or off. Many of the components of human milk enhance the function of metabolism or how genes are expressed. This field is relatively new, and research is ongoing in pursuit of all of the different complexities involved in it. Developmental programming, also referred to as fetal origins of adult disease or Barker hypothesis, is the basis for the observation that low birth weight is not only associated with immediate morbidities for the neonate, but also leads to later risk for adult diseases. The theory implies that there are critical time periods during fetal and postnatal development when an individual is sensitive to environmental stressors. During these early periods of plasticity, changes to an individual's metabolism can remain permanent. The adaptations that occur during critical periods of fetal and postnatal development promote survival in an inadequate environment, for example, poor nutrition or growth restriction. And so in these surviving individuals, later in life exposure to nutritional abundance and abundant growth, not necessarily healthy growth, but abundant growth, can cause metabolic disturbances that promote the development of diseases such as hypertension, obesity, and diabetes. And let's look quickly at stem cells. So stem cells have the remarkable potential to develop into many different cell types in the body during early life and growth. In addition, in many tissues, they survive as a sort of internal repair system, dividing essentially without limit to replenish other cells as long as the person or animal is still alive. When a stem cell divides, each new cell has the potential either to remain a stem cell or become another type of cell with a more specialized function, such as a muscle cell, a red blood cell, or a brain cell. Stem cells are distinguished from other cell types by two important characteristics. First, they are unspecialized cells capable of renewing themselves through cell division, sometimes after long periods of inactivity. Second, under certain physiologic or experimental conditions, they can be induced to become tissue or organ-specific cells with special functions. In some organs, such as the gut or bone marrow, stem cells regularly divide to repair and replace worn-out or damaged tissues. In other organs, however, such as the pancreas and the heart, stem cells only divide under special conditions. Until recently, scientists particularly worked with two kinds of stem cells from animals and humans: embryonic stem cells and non-embryonic somatic or adult stem cells. In 2007, researchers discovered stem cells in human milk, and to this day, this continues to be a very stimulating and progressive area of study. Investigations of late have found human milk stem cells migrate and assimilate into organs in utero and have importance operationally later in life. There is a possibility of mammary stem cells being reprogrammed for varied use, and there are ethical considerations involved. Is human milk sterile? Not at all. However, anti-infective agents maintain a low bacterial level for hours. This essentially means that the mother's milk can defeat pathogens in the infant's GI system before they can impact the baby. And recall that there are a number of factors in breast milk that coat the GI tract, making it difficult for invading organisms to invade. I have discussed the potential harm to an infant's GI system extensively, and however, human milk goes way beyond these illnesses. They also serve as protection against certain childhood cancers, as well as a myriad of chronic and autoimmune diseases. They also offer protection to the mom herself, decreasing the risk of premenopausal breast cancer. So we have already discussed other childhood illnesses like otitis media and diarrhea, and there are numerous tables listed in your text that contain other protective examples of human milk. These conditions and disease states include everything from alcoholism to schizophrenia, and these supporting studies are listed as well. The benefits of breastfeeding, even in our current limited but growing understanding, surpass artificial baby milk. To consider what future researchers will uncover, not just in the superiority of human milk nutritionally and functionally, but in the extreme weakness and toxicity of formula, is sobering. Researchers are interested in human milk, and nitrogen compounds top the list. Non-protein nitrogen compounds, beginning with nucleotides, are an essential ingredient to immunity. Nucleotides are critical to numerous physiological functions, including growth, development, and maintenance of the GI and immune systems, and nucleotides are the host defense against pathogenic attacks. Absent in cow's milk unless synthetically added, breast milk again provides the necessary components. Prostaglandins, bile salts, and epidermal growth factor are additional areas in which researchers consider of great consequence to the study of human milk. Why are formula-fed infants more susceptible to allergies and asthma? Formula-fed babies have less beneficial bacteria, they have greater pathogen exposure, and they have more exposure to harder to digest formula. Besides diarrhea and ear infections, what other disease states does breast milk protect infants from? So other disease states that breast milk offers protection from are certain childhood cancers, allergies, asthma, autoimmune disorders, and for mom, we should mention premenopausal breast cancer. So there are a number of anti-inflammatory and pharmacologically active components that we haven't discussed in this module, but that we should at least give mention to, and I believe that your text does cover some of these to a certain extent. There are prostaglandins, ovarian steroids, gonadotropics, somatostatin, prolactin, and insulin. Hormones are special chemical messengers in the body that are created in the endocrine glands, and these messengers control most major bodily functions, from simple basic needs like hunger to complex systems like reproduction and even the emotions and mood. And they are regulatory substances produced in an organism and transported in tissue fluids such as blood to stimulate specific cells or tissues into action. And more researchers are discovering and understanding the intricate role that they possess in human milk. To date, over 300 peptides have been identified, and we'll be looking briefly at the following hormones and their role in infant growth and development. Cholecystokinin, or CCK, is triggered by an infant's active suck. It is a digestive hormone found in the GI tract and brain, and production of CCK may be why mothers and babies experience a sense of sleepiness and relaxation during breastfeeding. So leptin is one of the hormones directly connected to body fat and obesity. Leptin, a hormone released from the fat cells located in adipose tissues, sends signals to the hypothalamus in the brain, and this particular hormone helps regulate and alter long-term food intake and energy expenditure, not just from one meal to the next, but again, that's long-term. The primary design of leptin is to help the body maintain its weight. Because it comes from fat cells, leptin amounts are directly connected to an individual's amount of body fat. If the individual adds body fat, leptin levels will increase. If an individual lowers body fat, the leptin level will decrease as well. Leptin helps inhibit hunger and regulate energy balance so that the body does not trigger hunger responses when it does not need energy. However, when levels of the hormone fall, which happens when an individual loses weight, the lower levels can trigger substantial increases in appetite and food cravings. This in turn can make weight loss more difficult. It assists in the formation of neural circuits that control food intake, energy intake, and weight for adults. In infancy, breast milk may contribute to helping infants regulate current intake as well. Current studies have seen that infants receiving formula have low levels of leptin and higher body mass indexes, or BMI, leading researchers to believe that the absence of leptin in formula feeding may contribute to formula feeding leading to obesity. Ghrelin is central to appetite normalcy and the release of growth hormone. Now, ghrelin is produced in the stomach and small intestine, with a little bit of the hormone released in the pancreas and the brain. It's been called the hunger hormone because of its role in controlling appetite, but that is just one of its functions. So it has many roles, but the one that it is most well known for is in the ability to stimulate appetite. It causes an individual to digest more food and store more fat. And in fact, when given artificially to humans, the amount of food intake can increase by 30%. So this hormone plays a role in the hypothalamus, which is the part of the brain that controls the appetite. And it may also act on regions of the brain that control reward processing. So it also plays a role in the pituitary gland's function, where ghrelin receptors trigger the stimulation of the hormone. It appears to help control insulin release and plays a protective role in cardiovascular health. This well-rounded hormone has a job in many different bodily systems. Ghrelin is higher in foremilk, and triglycerides and leptin are higher in hindmilk, and this may help babies register satiety and self-regulate. As artificial baby milk is unchanging or static through the feeding, there may be a disconnection with or delay in satiety, leading to a lack of self-regulation, which can lead to additional calories. Adiponectin. This hormone, secreted by adipose or fat tissue, is one of the cell signaling proteins found in human milk. It serves to regulate lipid and glucose metabolism, influence infant growth and development, function as an anti-inflammatory, and is inversely related to insulin resistance and fatness. Levels of this hormone decrease in breast milk over time and may possibly play a role in helping premature infants adapt metabolically. It may also assist with normal growth and development. Resistin. So resistin is a protein peptide in breast milk, and it's released by fat cells. This hormone is still a little bit of a mystery to researchers, but it may have a role in controlling the pace or speed of intrauterine growth. It may also support an infant's metabolic development. Now, obistatin. Newly found, obistatin is another peptide that may control sleep, boost pancreatic performance, and suppress appetite. So I won't even really cover this slide in depth. This is just another slide serving to highlight the many different complexities of immunological and nutritional constituents of breast milk. And you can see that there are so many other, constituents that we just don't have the time to cover in a 2-hour introduction to biochemistry of human milk. But there are other peptides that are involved in growth, energy, and maturation, in energy homeostasis and diabetes, as well as metabolic pathway regulation and postnatal organ growth and development. Other peptides are involved in the appetite and self-regulation of food consumption. And again, just the takeaway from these last slides are that, almost daily, research is published that highlights the remarkable properties of human milk, particularly metabolic programming and obesity. Studies continue to demonstrate the specificity and uniqueness of individual breast milk. So we'll finish this module with a comparison of human milk to manufactured milk. Human milk matches more than 50% of baby's genetic material. Milk from other species and plant-based fluids are genetically different from the human infant. Breast milk has a unique balance of nutrient and other components. It most closely matches milks of species with high maternal investment and frequent feedings. Human milk composition, as we've mentioned earlier, is dynamic. It's not static or uniform like artificial baby milk is. It is an evolution. Colostrum evolves to transition milk and then into mature milk. Colostrum in the early weeks is more accurately described as an immune layer that persists throughout the duration of lactation. Bioavailability is low for all other milks. Iron absorption is greater in human milk. Human milk has little residue and low solute load, and nutrients are utilized efficiently. Nutrients serve more than one purpose. Milk from other animals contains deficiencies and or excesses of one or more components, only some of which can be modified for infant consumption. Manufactured milk has 100% foreign proteins, and those are derived from ruminant animals such as cows or goats or plants. So formula can also increase the incidence of allergies, and cow's milk protein and soy are known triggers for diabetes in genetically susceptible infants. There can be decreased arousability in formula-fed infants, which has an implication in sudden infant death syndrome, and there may also be compromised heart rhythms. Now, bovine milk is high in phenylalanine, which we mentioned much earlier in the module, which can increase the complication of PKU, which is phenylketonuria, which is a metabolic disorder which can lead to developmental delay. Now, soy milk has many bioactive components, including phytoestrogens, aluminum, and magnesium, and research is still being done to see what the possible implications of those are. There are also non-human fats in formula with no capacity to protect the myelin sheath, heal from injury, or help to develop the central nervous system. With formula, there's also an increased risk of multiple sclerosis, altered cholesterol metabolism, and altered vision and cognitive development. There may also be the absence or deficiency of lactose, and lactose, as you'll remember, is a huge component of breast milk, and it's needed for a number of functions, but especially for the central nervous system and cognitive development. So with manufactured milk, there's also a lack of regulation, and some components in special formulas are unregulated, and there may be recalls for mistakes and even contamination at times. Powdered formulas are not sterile, and there may be manufacturing errors. There's also the experimental nature of a product, including additions of certain fatty acids like DHA and AA, and those are manufactured sources, and there's no real evidence to support the efficacy or safety of manufactured DHA and AA versus those from human milk. Formula feeding is also indicated in suboptimal growth patterns. Formula-fed children are fatter per length with smaller heads. They meet head circumference and other developmental milestones at later dates. There's an increased incidence of short-term illness, as well as an increased incidence of long-term and chronic illness. And there's also altered metabolism for women who do not lactate. So when you look at calories, usually they are fairly similar at around 20 calories an ounce, or about 60 to 70 calories per milliliter. And those are based on an average infant intake of about 750 milliliters a day. Now, this is a smaller range than the highly variable human milk. The protein content of certain formulas may be higher depending on the source of protein, which is usually based on the milk of cows or other animals or soy. As mentioned earlier, formula is more difficult for an infant to digest, primarily because of the higher levels of casein. Most formulas are modified to increase the weight to casein ratio, and while that may seem like that's a good thing, like that's a beneficial thing, remember that an infant's GI system is supposed to mature. It's supposed to be challenged in anticipation of the more difficult proteins that it's going to be, eating as it ages. Soy-based formulas are often used when infants are allergic to cow's milk, although frequently that doesn't solve the problem because many babies who are allergic to cow's milk are also allergic to soy. And soy formulas are also more commonly made without lactose, which, again, as we mentioned, lactose is an integral component for so many reasons of human breast milk. And even the lipids that are put in manufactured milk. The lipids are often vegetable oils, and in the past, they were even using trans fatty acids. And as we mentioned earlier, even though we have essential fatty acids and polyunsaturated fatty acids and long-chain fatty acids in manufactured milk, again, these are manufactured. They're not human-based. And so sometimes, even though it may seem like, well, we're putting in essential fatty acids, this is a good thing, there really isn't a way of knowing yet if that's actually beneficial due to the fact that it was manufactured and not innate. So finally, let's look at hydrolyzed formulas. So hydrolyzed formulas are specialized formulas, and they do offer complete nutrition for infants who can't digest intact cow's milk protein. They also help infants who are allergic to intact cow's milk protein. If an infant is allergic to cow's milk protein, these formulas will not be beneficial. And lactose-free formulas do not assist with a protein allergy, only with offering an alternative to lactose. But again, a lactose-free formula, it is very rare for infants to be lactose intolerant. The bottom line with comparing human milk to manufactured milk is that 50 years of technology will never beat a human breast. And this is just a quick summary of formula as compared to breast milk. And you can see all of the different additions that are being put in formula. Probiotics are added, prebiotics, minerals, vitamins, fats, DHA and AA or ARA, carbohydrates, protein, and water. And so, again, those are included. So it looks pretty complete. But then when you look over at human milk, the antibodies that are present, the anti-cancers, the growth factors, the enzymes, the disease-fighting stem cells, the hormones, the antivirus constituents, the anti-allergy constituents, the antiparasitics, with, again, probiotics, prebiotics, minerals, vitamins, fats, and especially the specific essential fatty acids from human milk, and again, the DHAs and ARAs, along with the carbohydrates and protein and water, there really is no comparison. Name 3 ways formula is different from breast milk. So 3 ways that formula is different from breast milk include genetic matching, the dynamic nature of breast milk, the evolutionary nature of breast milk, the bioavailability of breast milk constituents, the residue and low solute load of breast milk, the efficiency of breast milk, and just the nutrient composition of breast milk. Why do some moms believe that their babies do not get full or satisfied on breast milk? Breast milk is more easily digested and therefore more quickly digested. And what some moms may view as satisfying or filling is actually the baby's digestive system working much harder and much longer to digest the formula. So in 5 years, I am certain that we will be amazed at what we did not know about human milk and breastfeeding in 2018. The impressiveness of human milk, as well as the consequences of artificial milk, are far-reaching and expanding as research daily uncovers the unfolding beauty and power of human milk. As future lactation consultants, asserting that breastfeeding is not a lifestyle decision but an investment in health is key. A lack of breastfeeding support is one of the chief reasons why women do not breastfeed. This is a great quote by Dr. Ruth Peterson. Breastfeeding provides unmatched health benefits for babies and mothers. It is the clinical gold standard for infant feeding and nutrition, with breast milk uniquely tailored to meet the health needs of a growing baby. We must do more to create supportive and safe environments for mothers who choose to breastfeed. In the slides following, I have included a number, again, you've heard this before, of quality, reliable resources for you to refer to, and from government agencies, journals, universities, consumer groups, professional organizations, and volunteer groups. There are many, many places to obtain dependable information. And pregnancy and lactation, again, are milestones in a woman's life, and she may be more receptive to nutrition education in these instances. So being a positive influencer by being ready with sound nutrition suggestions will benefit mom, baby, the entire family, the community, and hopefully for generations to come.